Endocytosis is when cells take in substances by vesicle formation. Part of the plasma membrane invaginates to envelop the substance. The membrane then pinches off to form an intercellular vesicle. There are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is large particulate matter, such as food molecules, viruses, or even entire cells. This is most common in amoeba and microphages. Pinocytosis are usually liquids and small particles, and this happens most commonly in blood cells and plant root cells. We'll get more into receptor-mediated endocytosis later.
Exocytosis is a process by which cells move materials from within the cell into the extracellular fluid. In exocytosis, the vesicle fuses with the plasma membrane as secretion occurs. The vesicle membrane becomes part of the plasma membrane. Cells of particular organs are specialized to produce and export molecules. Pancreatic cells release insulin and enzymes, and anterior pituitary cells release growth hormones.
Receptor-mediated endocytosis is a form of endocytosis where receptor proteins on the cell surface are used to capture specific molecules. The receptors, which are transmembrane proteins, cluster in regions of the plasma membrane known as coated pits. This name comes from a layer of proteins called coat proteins that are found on the cytoplasmic side of the pit. When the receptors bind to their specific molecule, endocytosis is triggered. The receptors and their attached molecules are taken into the cell through a vesicle. The coproteins are used in this process by giving the vesicle its rounded shape and helping it bud off from the membrane. Receptor-mediated endocytosis allows cells to take up large amounts of molecules that are relatively rare in the extracellular fluid. Although receptor-mediated endocytosis is intended to bring useful substances into the cell, it does at times bring in less friendly particles that may gain entry through the same gate. These include flu viruses, diphtheria, and cholera toxin, as they all use receptor-mediated endocytosis pathways to gain entry into the cells.
Active transport is the process of moving molecules across a cellular membrane through the use of cellular energy. In active transport, carrier proteins, often called pumps, and energy, usually ATP, is required. These proteins transport molecules against their concentration gradient. Carrier proteins use energy to move molecules across the concentration gradient. And an example of active transport is the sodium-potassium pump, which is especially important for the nerve and muscle cells. It maintains cell membrane potential by pumping 3 sodium ions out of the cell for every 2 potassium ions that move into the cell. The carrier changes shape after the phosphate donated by ATP attaches, and then again after it detaches.
Facilitated transport is one way of moving those materials without expending cellular energy. It explains the passage of molecules such as glucose or amino acids. In facilitated transport, materials are moving down a concentration gradient. In other words, they are moving from an area of high concentration to low concentration, as in passive diffusion. However, the presence of the cell membrane prevents passive diffusion from moving the materials easily, because a protein-free lipid bilayer is highly impermeable to ions. Small nonpolar molecules like O2 and CO2 are soluble in lipids and diffuse rapidly across lipid bilayer membranes. Uncharged polar molecules such as water and urea also diffuse across the bilayer, but at a much slower rate. Charged molecules, however, do not readily diffuse across a lipid bilayer membrane, and they are required to have additional assistance. In facilitated transport, membrane proteins assist diffusion of materials through the cell membrane. The protein binds the molecule being transported on the surface of the membrane that passes it into the interior proteins that typically form a channel or pore in the membrane. These proteins are called transport proteins. One type of transport protein is known as a carrier protein, where the target molecule to be transported binds to it it undergoes a conformational change, which moves the molecule from one side of the membrane to the other. There is also glucose transporter 1. There is also glucose transporter 1 protein, which is an example of a carrier protein for facilitated transport of glucose into cells. The other category of transport proteins is called a channel. A channel protein has hydrophilic portions on the inside and outside of the membrane, and a hydrophobic portion that penetrates the membrane in order to form a channel. An example of this will be aquaporins.